today I'm going to show you how to draw blood from a jugular vein. So these are the materials that you need. You need a syringe. We use our 3 ml syringes. And then you have 2 different types of blood tubes, a red top that has the serum separator and a purple top, the EDTA tube that has the heparin in it to keep the blood from clotting. Then you also have alcohol to spray to part the fur to find the vein. So I'm going to hold off in the jugular groove, and then I'm going to spray the fur with alcohol. I'm going to separate the fur and fill for the vein. As soon as I fill the vein, I'm going to uncap my needle, and with the bevel up, I'm going to insert and draw the blood. And then as soon as I'm done, my holder is going to hold off, and then I'm going to insert the blood into the tubes. You put about 1 ml into the purple top, and then you invert it to prevent from clotting, and you put the rest of the blood into the red top. Okay, so today we're going to be doing our buccal mucosa bleeding time. So the things you need for that are your stopwatch, your lancet, and your filter paper. And what we're going to do is we're going to measure every 5 seconds we're going to blot the spot that's bleeding until it stops bleeding, and that's going to be your clotting time. So the first thing we're going to do is hold the lid back. We're going to right above the tooth, and then we'll start our timer. And every 5 seconds we will blot. And you just keep blotting until she stops bleeding. Alright, so she has stopped, so her buccal mucosal bleeding time is going to be 47 seconds, and that is normal for our dog, and that is how we do our buccal mucosal bleeding time. So today I'm going to show you how to make a blood smear to look under the microscope to check for all the white blood cells in a blood smear. So the materials you're going to need is you're going to need your blood sample. It needs to be in the EDTA tube or the purple top tube. And then you're going to need a pipette because you only need 1 drop of blood. And then you're going to need 2 separate slides, one to make the smear and then one for the blood to go on to push. So first you're going to open your blood tube and get 1 drop of blood. So you only need 1 drop of blood on your slide. And then you're going to hold your slide with the one hand, and with your other hand at an angle, you're going to push your slide all the way back to where the blood completely fills the bottom of the slide, and then push forward. And then you have made a blood smear, and now you're going to let it dry, and once it finishes drying, you will stain it and then look at it under the microscope. So once you've completed your blood smear, it's had time to dry. You need it to make sure it's completely dry before you can stain it. So this is my completed blood smear. You can see the nice feathered edge towards the top. And then you also want to make sure you label it with the date and the canine or feline's name on it so you know whose blood smear it is. So the things you need for the blood smear to stain it are going to be your 3 stains. You also need distilled water because you need to rinse it off before you look at it under the microscope. So what we're going to do first is open all 3 of our stains. And then you leave it in each stain for about 10 seconds, or you can dip it 10 times. So the first thing we're going to do is leave our first one in for about 10 seconds, and you just completely submerge it and let it sit in there. That one fixes the stain fixes the blood smear to the slide. And then after that, you put it into our second one. And you do each of these for 10 seconds. Once they're completely done, we're going to rinse the back of our slide with distilled water. That's what I have in this cup right here. And then once our slide is completely dry, we will look at it under the microscope. You start at the lower power and you work your way up. you are going to end up looking at under your microscope on the oil immersion lens so you can see your white blood cells. And after each dip, you just kind of wipe off the excess off the side of the slide. And then you're going to take it and you can hold it over the sink. And then you're just going to rinse the back of the slide with the distilled water. And that's just to get all of the excess stain off of the slide. And after that, you can use a paper towel and kind of wipe off the end of the slide. Then you're going to let your slide dry upwards so all of the excess water and everything goes down and does not affect your feathered edge. So once your slide has been stained and it's fully dry, you're going to place it onto the microscope. With a blood smear, you need to start using the 10x lens, and then you scroll over to the feathered edge. And once you notice and you can see all of the red blood cells on there and any of the white blood cells, you are not going to see until you are fully in the oil immersion lens. Once you get it focused, you can move your lens. You can put 1 drop of the immersion oil on the section you're going to be counting. Make sure you only use the oil with the oil immersion lens. It can actually mess up the other lenses if you use that. So just make sure you clean your microscope lenses really well after you finish your smear. So once you have the oil on, you have to refocus your slide, and you'll see a bunch of the red cells first. And once you start getting it all focused, you just scan the slide. To read a blood smear, you have to count to 100 all of the different white blood cells you see. So you will see lymphocytes and neutrophils mostly, but you may also see some basophils, some eosinophils, and you might also see some bands. And then you will also see platelets and some red cell morphology, and these are all the types of things that you need to record when you're doing a manual differential. And that's how you read a blood smear. So today I'm going to be showing you how to run a blood chemistry test on one of our patients for their annual blood work. So the first thing we're going to do is pull them up in our system, which we use Avamark. So we're going to make sure we have the patient up so the blood work saves into the computer. Once we have the patient up, we are going to make sure we have a blood sample, which we do, and we spun it down. We're going to use the serum for this sample. So the first thing you need is a rotor. So we're going to open the rotor. You also need to have a pipette and pipette tips. Get that, and then you're going to open the blood tube. When you press down on the pipette tip, it pulls up the correct amount that you need for the rotor. You just don't touch the bottom of the tube. So you're going to put the pipette in and draw up the blood. And the next thing on the rotor, it's hard to see, but there is a circle where you're going to insert the blood. So once you insert it, you do it in one swift motion because you want no air bubbles. Once that is completed, you open the machine. You're going to place the rotor and hit close. Next, you're going to choose the species, so we have a dog today, and then you're going to enter their patient ID. And then you're going to hit done. And then it will analyze, and as soon as it's done analyzing, the results will print out and save to the computer. So once the analysis is complete, you make sure the results have been printed. They automatically save to the computer, so once they've been printed, you can pull up the results and share it off. And then here are the results that we'll give to the doctor to read to see how the patient is doing. So today we're going to be doing a pancreatitis snap test or CPLI test on a dog that hasn't been eating and not drinking and shaking for about a day and a half. So the first thing we need to get is blood off the dog, and then we're going to take 3 drops of this blood. You put 3 drops into the tube. 2, and then you use 4 drops of the conjugate. 2, 3, 4. And the next thing you're going to do is invert the tube 3 to 5 times to mix the blood and the conjugate together. And then next what you do is you pour the whole tube onto this section of the snap test. And then what we're going to do is we're going to wait until the blood and the conjugate reach down to this part of the snap test, and then we will snap it. Once it reaches the bottom part and you snap it, you will wait 10 minutes for the test results, and then you will check to see if it's going to be a positive or a negative. Right before we snap it, we're going to move it right over here. Now we are going to snap it. So our pancreatitis test is done running. It's been 10 minutes. So when you look at the test, we'll do a close-up now. The dot on the left side, the darker dot, shows that the test is normal, and the other dot is a lighter color, so that means that it's normal. If it was abnormal, the other dot would be a darker color, so our patient does not have pancreatitis. So I'm going to be showing you how to do a PCV-TS test, which stands for packed cell volume and total solids. So we're doing this to see the amount of red blood cells, and then we'll use the plasma and our refractometer to read the total solids. So the materials that you need for this is you need your blood sample in the EDTA tube, so it doesn't clot. You're going to need the microchromatic tubes. We have 2 different sizes, so you can use the little ones or you can use the big ones. You're going to need some clay to stop the end of your tube, and then you need a refractometer because this is what you're going to use to read the plasma once the blood is done spinning. So the first thing I'm going to do is going to fill these tubes up. I'm going to use 2 of them at the same time so I can balance out when I centrifuge them. They have these little red lines. You can have those at the top, and then you just open your blood sample and tilt it and they will fill up. And you can do them both at the same time, so you have about the same amount of blood in there, so the centrifuge will be balanced. So once they are both filled with blood, you're going to take them and press them into the clay. This is going to stop the blood from spinning out when you centrifuge it. So I'll kind of show you this. That's where the clay is at the very bottom, and that's going to keep the blood from spilling out while you centrifuge. So next thing you're going to do, so once you have the clay stoppers in, you're going to insert with the clay up, and you're going to insert your tubes, 1 on either side, so you have it balanced for the centrifuge. The next thing we're going to do is we're going to put this into the centrifuge, and according to whichever centrifuge directions yours has to spin the blood, that's how you're going to spin them. And ours we spin on the 30 speed for 10 minutes. So our blood sample has finished spinning, so now we're going to remove it from the centrifuge. And just be very careful with these tubes because they are glass. So we're going to pull them out, and we're going to set them. This is the reader that comes with the microhematocrit tubes. So to read these tubes, what you're going to do is, so I can show up close, this is the plasma and this is the blood. So what you do is line it up with the 0, and then you just move it until it meets a line. So this one is right at 40. So our PCV for this blood sample is going to be 40. So the next thing you need to do is you're going to read it in a refractometer. So you just read the plasma for this portion. So what you need to do is carefully break your tube. Just make sure you don't get any glass on the tube. So you just kind of break it, and you want to break it above the buffer line. And then you're going to just gently tap it on here until you get the plasma to come out. If you don't get any to come out, you can always break your tube a little more. You don't need very much. So we're going to break our tube just a little bit more. So then you can just get the plasma out on your refractometer. Okay, and after it's finished, you are just going to use your refractometer, and you need to look into a strong light source, and when you look in, you will get your results from that. You just look, and then you focus it. So it looks like our reading is going to be at 3.02. And that's how you read the plasma.